see how this works. Oh yeah, so I have no conflicts of interest to declare. So I know we're all ready for coffee and biscuits in a moment, but the whole point of this session, and I hope it's become really clear, is to see the breadth of research that is possible by the components that we make. And you know, the idea is to really step through the looking glass and not only think sometimes what's the important research from a transfusion and hematology point of view, but what can be accomplished with the components that we have. So I put down that quote there, one man's trash is another man's treasure, because really the availability of our buffy coats especially is so important for so many aspects of research. So what is the contribution at the moment? We actually have 105 third-level researchers that are approved to use our products to progress their research all across Ireland. As you can see here, the majority of these individuals really want our buffy coats, but there still is lots of other products that are requested over time, and sometimes different groups would like to have matched plasma and buffy coats and things like that. So this has really led to us having, a number of kind of beyond this research collaboration agreements. And at the moment we have those with 6 researchers in 4 different universities and 2 national hospitals. So this is just an example of a couple of the institutions that regularly access our products. So I'm missing a slide, but anyway, what I want to talk to you really about today is, first of all, it may sound really simple, but where do the components come from? Putting the donor back in the center of this process. So, as a researcher, if you are looking to access our components, what has to happen before they come to you? And then give you, I suppose, a flavor of the various and really, multidisciplinary projects that are ongoing at the moment using our products. And I suppose, what is the process around approval for that, and where can we go from here? So it might sound really simple, but blood components come from blood donors. And the reason I'm saying that is there are many researchers that, for us, the blood component production is almost that last point in the process before it goes to the hospital for a transfusion. But obviously for so many researchers, it's their starting material, their starting point. So this slide here is just to really make clear as to the whole process beforehand. And so if you just think about what has happened before you've started using our products, there's a donor has had to come in, they've had to qualify, they may go through a rather personal questionnaire. What was their motivation for donating? It's unlikely that it was initially, I'm going to go and donate just to give my product to research. They probably, they want to help patients more than likely first and foremost. They've had to go in and had their blood drawn. This takes time. Maybe they were phoned because there was, an urgent need for their particular blood group. There's different types of collection. So do they have to come and travel maybe to one of the fixed apheresis clinics, or is it whole blood donation? And then their sample has to go for testing to a number of laboratories. So we've got blood grouping laboratory, infectious disease screening, and then on occasion genotyping, depending on the process required. And it's at that point then that we make the blood components. So I think it's really important to think that, we have a duty of care to our donors, that their motivation to come in and donate, many I think are so happy to be involved in research, but that's unlikely to be the initial reason they engage with our service. But it is true that some donated products cannot be transfused, and some products that we make, many of which being the buffy coats, are kind of byproducts of the process itself. And by providing our blood components for research, we are able to maximize, the utility of all the donations that we have. So it's a really important facility that we offer. The donors themselves, find out a little bit about this process, their donations, if it can't be transfused, through the donor booklet. They sign a donor declaration in the knowledge that the anonymous donation components may go to third level research where possible, but we've recently now published a much more detailed IBTS research and development donor research booklet, which goes into, I suppose, a lot more around what might happen to your component and what might we use it for from a research and development point of view. And actually out on the Give Blood stand out there at the moment, there are copies of this if anybody is interested. So it is really true that we have a sustainable supply of leukocytes for immunological research. Gone are the days where we used to bleed each other in the lab. I know when I was doing my PhD, we were always delighted to have a medic doing their MD because we would make them bleed us so we could isolate PBMCs, and that's what we worked on. I think we can all agree that it's good that we've moved on from that. And you know, it is all about maximizing the contribution of the donor, but at the same time remembering that we're not merely supplying a product. This has come from somebody, and that we have to really keep them central in our mind. I did a bit of a word cloud as to all the various kind of fields that the research beyond our doors happens, and you can see very clearly that immunology is shining out there as a main one, but it's not the only one. So there is, you know, studies ongoing looking at disease association, signaling, cancer, metabolomics, hemorrhage, cell therapy. So sometimes, you know, we don't often know the various different research that is ongoing using our products. And I think this is a whole idea of this session, was to really have a look and see, I suppose, what can these products really be used for. So the system at the moment for application and approval, for those of you that actually use our products, this is now going online, and we'll be having a Zoom with you in the next little while, informing you all. But it's kind of a three-step process. So the researcher themselves has their, you know, research question. We will now be kind of asking, you know, upfront for your ethics information, because obviously we can't ethically approve your research. We can certainly help you along the ethics journey, but it's very important that it's at that researcher point that's given, and the kind of products that you're looking for. They generally then come to me to review. And we have to kind of considerations here around all the data protection regulations, the health research regulations, and most of the time people are happy with kind of anonymous blood components for their work. And it goes for approval. The new system will be asking for acknowledgement to the IBTS for providing this, and I think that's really important so that donors themselves can see how, you know, their products are being used in various research. And then there are times when we'll have further follow-up. So we need to know if there's going to be donor-related research happening. You know, are you trying to find out something about healthy donor populations? Maybe you're looking for age and sex as well, along with the components. And that is important that we maintain oversight on donor-related research, but also we're delighted to be involved. We just might need a little bit more information. Then I may be knocking on your door if they're working in a field allied with transfusion and hematology, if it's, you know, really closely linked with what we want to do from our own R&D strategy. Then again, we can kind of go on and build further research collaborations. And I'm just going to kind of briefly talk through a couple of different projects where I think we have used those relationships now over the last few years to really kind of capitalize on what was initially very much a researcher, you know, using our products. We kind of wouldn't have had a lot of interaction into really building research collaboration and, you know, longer term projects and closer relationships. The first is a PhD project that we will be starting in January. This is co-supervised by Dr. Claire Wynne and Fabian McGrath and myself. Fabian and Claire are based in Technical University Dublin. TU Dublin is where so many of our medical scientists get trained, so they have been like long-standing users of our components. But I suppose when reaching out and talking about, you know, their own expertise and Claire Wynne's expertise in extracellular vesicles, our own need to really keep up our skills and develop them further in blood components, meant that we have a project now which is going to ask the question, you know, what are the molecular changes that occur in platelet pack-associated EVs, and how can these be exploited to improve quality and reduce waste. We want to define all these kind of characteristic and functional changes that are going, and see during preparation, processing, and may, I suppose, ask the question then, you know, what is there any sort of donor variability that is impacting this as well? Another, and was mentioned there by Professor Doherty, we have now a much closer relationship with James's and Trinity through the Cellular Therapy Research Group. I think we really, you know, understood that firstly, with so many of our research products going out for immunology, we need to learn about this. But secondly, cellular therapy is such an important and growing area that, you know, through those links, again, these laboratories that kind of have routinely used these products for a long time, by developing a much closer knit collaboration, we could really be involved. And by doing that, that means, you know, we can provide a lot more information than you would kind of get, or maybe other products than you might get. So Hayley's poster was up yesterday, and we're delighted to be involved in that project too. And then Dr. Ingmar Schön has just presented there on, I suppose, a growing collaboration now between RCSI, Vascular Biology Group, and the IBTS. And, you know, we've kind of tried to combine our two areas here of expertise and the availability of platelets that we have, and I suppose the access that we have to donors to maybe consent them for research, so that we can really look at the impact of storage conditions on platelet function and clearance, and look at the effects of ABO expression in relation to major hemorrhage. And as Ingmar has just said, we do have 2 postdoc positions that are currently advertised, so we would be delighted to get any information, or anyone who wants to talk about that, please come find us. One of the things that I was chatting to a few people yesterday, and delighted to see so many different individuals from across the IBTS here, is that the actual collections teams maybe could be really involved in some of this research, because it is at that point that we have access to our donors, and may need to consent donors and kind of teach them about research, so that it's not just in the laboratories or the analysis after the fact. It really is about kind of reaching across the whole service. I'm now going to give a really high level just kind of list of the various research projects that are ongoing, and that's just to really bring home, I suppose, the widespread nature of all the research that is happening. So, you know, there are projects looking at novel therapeutics for the treatment of chronic inflammatory diseases, the impact of oncology treatment on subsets of immune cells, and innate immune signaling and metabolomics. Investigating the molecular mechanisms of T cells, activation and migration, and the general role of lymphocytes in health and disease. Investigating age and sex differences in innate immune responses. I think it's really important here, I mentioned method validation in teaching. So again, if you are in a hospital laboratory or an undergrad student, maybe trained to be a medical scientist or in various areas, we also provide products for that teaching and training, and maybe for comparative and diagnostic purposes as well. Bioengineering, so developing a humanized model for myocardial infarction. This is really interesting. This is with the engineering department in Trinity, the first time they've really ever accessed our products. And I was kind of amazed to see, you know, the difference in what can be done. We have gone into a collaboration agreement with the IPATH study, which is looking at immunological markers of arthropathy in individuals with haemophilia A, and again, the analysis of innate immune parameters, providing the healthy controls for this study. Vaccine development for Staph aureus infection, the role of exosomes in lymphoma, T-cell signaling in retroviral infection. And also, I'm one of the poster winners here, accurate blood loss assessment, looking at developing a tool for this during labour and delivery, so providing whole blood plaques to really, you know, develop a novel way of assessing this that is directly, you know, clinically applicable to mothers during delivery. So, oh, I was wondering what happened. Here we go. Okay. So really, the take-home message of this whole session, and, you know, this kind of high-level talk here, is our IBTS research extends far beyond our own doors. It's really important that we can maintain oversight and always meet the duty of care to our donors first and foremost. But at the same time, with that in mind, we're really excited to kind of build on these relationships, capitalize on, you know, so many researchers that have used our products for so long, and kind of ask those questions. Are there kind of further collaborations we can do? How else can we help make those projects better? I think before I finish, it's so important in this session on components for research that I acknowledge all the donors. Without them, we would have none of these components, and it is through their agreement that we are able to provide them for research purposes. To all the researchers and collaborators, without you, I would not have a talk today. I was able to look through, I couldn't believe there was 105 people that are already approved for accessing our products. And obviously to the IBTS, because again, you are producing these products on a daily basis, you're liaising with all the researchers and making that available to them. So thank you very much.
Thank you, Allison. I'm sure you're all dying for some coffee or tea. But if there's any questions you have for Allison, there's one over there. And I'm pleased to see it's Sean.
Hi. Hi, Allison. It's great that you put this conference on and you've presented to everyone today. As being in a very similar position in the Welsh Blood Service as you are in the Irish Blood Service, I congratulate you, because to say to the audience here, you are in a position of tension between the aspirations of researchers and the care of blood donors presenting for donation, and being the line of communication between the donors, but also mindful of the many legal governance, organisational governance and health care governance legislation that you probably have to adhere to is there. But I think positions like ourselves are paramount to having a professional approach to research and being that interface. But I just want to say, I know it's an extremely difficult role to sit in, the middleman, so I congratulate you for your progress here. You talked about the importance of donors and donor considerations. We did a very small survey with our donors, and we had one of our metrics was 85% of our donors expect their products to be used for research anyway. And going into a new phase, the importance of public and patient involvement, and in our case, donor involvement. So my question is, one thing I'm considering is whether we need more direct representation of patients and donors on our research committees, on our research management committees, and whether you're considering that.
Yeah, I completely agree. First of all, thank you very much, Sean. Yeah, but I totally agree. We actually had a meeting just before we came down here, just even talking about maybe the need for donor focus groups to find out, they're, reading their information booklets and they're signing, but how aware are they of the various kind of research that goes on, and to find out their awareness on that. But then also what we're assuming is going to be probably, they're delighted, they're already coming in to give blood. Most people that do that are very open to the types of research that goes on, but yeah, to kind of have those groups so that we can assess that properly. And I think it would be great to have better donor representation on R&D. I think it'll only helpful going forward, yeah.
I might add that I appointed both Sean and Allison to their jobs.
Thank you, Steve.
Right, any further questions? Yeah, there's somebody over there.
Just about the fact that all these products that are going out, would there be categorizations based on the studies on if you would give them like expired versus in-date products and stuff like that? Is there like a priority scale of, okay, this is technically unusable, we'll give that to donation, or this is just expired, we'll give that to research, or this needs fresh donations?
So I suppose, yeah, first and foremost, the priority is to meet the needs of a patient. Yeah. So, and then it's more about, if we have approved a researcher for access to particular components, we will be checking with the component laboratory that is feasible, and then they will kind of liaise directly with dispatch and request them themselves. So sometimes it is expired products. A lot of the time, as I said, it's buffy coats, which we can't use anyway. I think there is scope for a lot of other products there that, such as expired products, it's much better to, as I said, maximise their use than be discarding them. But at the moment, it is that kind of more simple process where we approve what they're looking for, and then patient first, and then if it's there, they will get the access.
Anything
Allison,
further?
hi, sorry, I just want to ask you a question in relation to research products and recalled blood components. Do you think we've got a responsibility to tell the research facilities if a product's been recalled in relation to illness or anything like that? Or what do you, like, where do you stand on that?
Yeah, so although they're anonymous, we still maintain a code that is, that we know every research facility that has been given products, so that if there is any need to contact them for anything that has flagged up, that we have the ability to do that. Yeah.